Okay, so everyone can see my slides okay?
We can see them.
Okay. Yeah, so thank you all for attending my dissertation defense, which is titled Computational 3D Optical Imaging Using Wavevector Diversity. Let me switch to a laser pointer. Okay, so we have a lot to get through, so let's get started. So I'll start with the formal degree requirements. So I've completed all the coursework required by the BME department. These courses have already been validated by the committee previously. So unless there are any objections, I'll just continue. Likewise, I've completed the RCR training and my 2-semester TA requirement, and these have also been previously validated by the committee, so I will continue. Here's a list of awards I've received during my PhD. I'll note that a substantial portion of my research was funded by an NSF graduate research fellowship. So here's a list of peer-reviewed publications that I've co-authored during my time at Duke, listed in chronological order. So of these 12 publications, I'm the first author on the 5 highlighted ones, some of which I'll talk about in this presentation. And in addition, here are 4 more papers that are in review or in preparation, on 2 of which I will be the first author. So of these 7 first author papers that are the subject of my dissertation, I will just be presenting on 5 of them, and I'll note them again as I cover them in this presentation. Here's a list of conference presentations and invited seminars on which I'm a co-author. And I have 4 pending patent applications related to this work, 2 of which are directly related to the work in this presentation. Okay, so now we get to the actual interesting part, the research. So here's an overview of the rest of my presentation. So I will lead with a unified theory of optical coherence tomography, or OCT, based on a case-based formalism. And this theory will directly relate to the title of my dissertation, as the idea of using wavevector diversity for 3D imaging will feature prominently in this portion of my presentation, well, and I guess the whole presentation. So this theoretical description will lay the foundations for the rest of my talk, in which I'll present a new class of techniques I developed at Duke, which is centered around incorporating extreme angular diversity in OCT. So we'll start with the unified case-based theory of OCT. So I'll note that this work has led to a rather unusual 50-page publication that recently got accepted a couple weeks ago by Advances in Optics and Photonics. So obviously 50 pages is too much to cover in this presentation, so I'll just present a few highlights. And if you're interested in learning more, I'll refer you to this paper once it gets published, which should be within a few weeks now. Okay, so since the inception of OCT now 3 decades ago in 1991, numerous forms and extensions of OCT have been developed as methods for obtaining depth-resolved cross-sectional images for various applications such as ophthalmology, cardiology, and dermatology, to name a few applications. So point scanning OCT is perhaps the most common form of OCT, and it's very successful as a clinical standard. However, people typically use weakly focused beams to get long depths of focus, such that the axial or Z resolution is often better than the lateral resolution, as is the case in these retinal images. In full field OCT, typically high numerical aperture focusing is employed to get these very nice high resolution en face or XY images of the retina. But as a result, they often trade off the depth of focus. So to address this trade-off, various extensions have been developed, one of which is a technique called interferometric synthetic aperture microscopy, or ISAM, which corrects for this focus computationally, but typically requires a nanometer scale mechanical stability, referred to as phase stability. So more recently, our group has developed a new extension called Optical Coherence Refraction Tomography, or OCRT, which addresses the same issue in a different way and without the stability requirements of ISAM. And you'll see more of this later in this presentation. So the motivation of this work is to advance a theory that unifies all of these major variants of OCT under a common framework of coherent imaging. And in doing so, we will be able to explain pretty much all the fundamental properties and concepts of OCT, in particular, the resolution, the contrast mechanism, and the trade-off between the lateral resolution and depth of focus that I just mentioned, Speckle, and the effects of dispersion and aberrations. So that's the motivation. But before we dive deeper into K-space theory, let's start with the fundamentals. So what is a K-vector or a wave vector? So K-vector and wave vector mean the same thing. And they refer to 3D vectors with components kx, ky, kz that compactly describe a plane wave. So a plane wave is a theoretical construct that describes a 3D wave with infinite planar wavefronts that extends everywhere in space and has uniform amplitude everywhere. So as we learned in, I guess, middle school or high school, vectors are characterized by a direction and magnitude. So the direction corresponds to the direction in which this plane wave is propagating. And the length of the magnitude, or the length of the vector, is called the wave number, denoted as k naught, which is inversely related to the wavelength lambda naught. We're using angular units in 2 pi over lambda naught. So the k vector is orthogonal to the wavefronts of the plane wave that it represents. And this is the full form of the plane wave. So this vector is just a compact wave representing this. So in subsequent slides, I'll often draw these parallel plane waves perpendicular to the vector just to remind us that these vectors are representing plane waves. Okay, so now that we've defined the k vector or wave vector, we now describe the Fourier diffraction theorem, or the fundamental theorem on which this whole K-space theory rests. So let's say we have this 3D sample, such as this cell, and we want to perform a 3D reconstruction of it, or in other words, obtain some 3D map of some intrinsic property of the sample, and in this case, the refractive index. I only do 2 axes here, the z-axis and the x-axis, but this argument applies to 3D as well. So you can imagine the y-axis pointing into your screen. It's perhaps easiest to understand this theorem in terms of, well, in the Fourier domain, which in this case is synonymous with K-space. So that's how this gets its name, K-space theory. And since there's a one-to-one relationship between real space and K-space, if you can measure everything in K-space, then you basically also measured the 3D sample. Okay, so let's say we illuminate this sample with a particular plane wave with some wavelength and some direction. So the sample will potentially scatter light in every direction depending on its structure. And now suppose we place imaging systems in every direction. Let's assume we cover every direction over 4 pi steradians with no gaps, and that we're measuring the field, so both the amplitude and the phase. The question that the Fourier diffraction theorem answers is, what information about the sample have we just measured? It turns out not that much, I mean, a good amount, but not that much. So according to the Fourier diffraction theorem, given this geometry, we measure, assuming that the sample is weakly scattering, we only measure information along this spherical shell known as the Ewald sphere. And the radius and position of the sphere depends on the illumination wave vector. So in particular, it's the reverse. So this vector I drew here is in the opposite direction of this vector. And just to clarify, I don't mean the information contained inside the sphere. I literally mean only the information along this infinitely thin spherical shell. So how do we get more information? Well, we're already measuring the field from every angle. So the other thing we can try is to vary the illumination angle. So for example, if we illuminate from this direction, the Fourier diffraction theorem tells us that we will measure this Ewald sphere as dictated by the illumination wave vector. So these are pointing in the opposite direction. Likewise, we can illuminate from a few more directions and obtain more information along these rotated Ewald spheres, so on and so forth. And in the limit, when we've illuminated from every possible direction and imaged from, we illuminate from every possible direction and image from every possible direction, all 4 pi steradians, we can theoretically measure everything in this 3D ball of radius 2k naught, or twice the radius of the Ewald sphere. So if you do this, you've basically measured everything in K-space, and therefore you have measured the sample's 3D refractive index distribution up to the diffraction limit. So this is basically the dream imaging system. This is the most information that you can ever hope to measure, barring some additional clever tricks or insights in super resolution microscopy that won the Nobel Prize in, I think, 2014. However, today, no practical system, as far as I'm aware of, has demonstrated illumination from every possible direction and a collection from every possible direction. So in practice, we can't do that. So we have a limited collection range, and we can only capture part of the Ewald sphere. So this part here corresponding to the angular coverage of your imaging system. Similarly, it's very difficult to illuminate from all directions. So you're often restricted to some cone, or restricted by some numerical aperture. And this means you only cover these shifted spherical caps in K-space. You can sample densely within this cone, and then you trace out this continuous bow tie shaped region, much less than that giant spherical ball that we saw a few slides ago, but still respectable. Another imaging geometry involves illuminating and collecting the sample from the same side. And this one is highly relevant for OCT as a reflection mode imaging system. So here again, the Fourier diffraction theorem tells us that we measure a partial spherical cap up here. So since the wave vector or K vector has both magnitude and direction, and we've already varied the angle, we can also try varying the magnitude, or basically using multiple wavelengths. So if we do that, then we get these spherical caps of different radii, depending on the length of this K vector. And we can sample, we can sweep densely within this bandwidth, and then we'll sweep out a continuous region like so. So still nowhere close to the spherical ball we saw earlier, but still very respectable. And this is what, in fact, what full field OCT measures, as we will see. So here's the Fourier diffraction theorem in equation form for those more mathematically inclined. So the sample is actually represented by the 3D scattering potential, so V of X, Y, Z. And this is directly related to the refractive index distribution via this one-to-one relationship. And then V twiddle is the scattering potential spectrum. So this is that K-space plot that I showed earlier. And then U is the scattered field that we measured along this Ewald sphere. And finally, the K illumination is the illumination K vector that I showed in previous slides. And note that there's a negative sign here, which explains why we have been reversing this vector when determining the position of the Ewald sphere. Just to reiterate, this is the fundamental theorem on which the rest of this part of my talk rests. So now that we've explained the Fourier diffraction theorem, let's take a closer look at the transfer function of full field OCT. So here we're being more physically realistic by assuming a Gaussian source spectrum with some center wave number K naught and some width sigma K. And we're also assuming a Gaussian beam with some numerical aperture, or NA, dictated by some angular range here denoted by sigma theta. I note here that we're expressing the K-space coordinates in spherical coordinates because of the symmetry, as it's symmetric around the KZ axis. So this is just a fancy equation that characterizes the shape that we saw earlier for full field OCT, but this time using Gaussian assumptions, we get these sort of plots on the right. We have a Gaussian decay along the edges. So I've simulated for a high NA and a low NA case. So the red and blue circles correspond to the Ewald spheres, where the wave number is corresponding to the full width half max of the source. And the wedges likewise correspond to the full width half max angular range, angular coverage of the objective's numerical aperture. So this transfer function is the product of two factors. As you can see here, one is roughly the axial component, and the other is the lateral component. And you only have separability between the axial and lateral resolution in the low NA limit, because note that this factor also has a theta dependence. So in the low NA limit, then this transfer function looks like the product of two orthogonal Gaussians, but in the high NA, that is no longer the case. So now we discuss point scanning OCT, which is perhaps the most common form of OCT. So point scanning OCT uses a scanning focused beam, and a focused beam is nothing more than a weighted superposition of plane waves. So for example, a Gaussian beam can be decomposed into a set of angularly varying plane waves, whose amplitudes are described by the Fourier spectrum, or also known as the angular spectrum, which for a Gaussian beam is not surprisingly a Gaussian. So the spectrum tells us the amplitude and phase of a plane wave traveling at a particular direction, kx, ky, corresponding to k illumination. So to generate the overall transfer function of the point scanning OCT system, we perform a weighted sum of full field OCT transfer functions for each illumination direction, as depicted here. So these are the full field OCT transfer functions for various illumination directions. And therefore, we can derive the transfer function of point scanning OCT. But to better appreciate its shape, we put it into the context of related imaging systems. So in the upper left, we have a monochromatic wide field microscopy, which is just an infinitely thin restricted shell, which is the Ewald sphere. In the upper right, we have the full field OCT transfer function we derived earlier. In the lower left, we have monochromatic reflection confocal microscopy. And then in the bottom right, we have the point scanning OCT transfer function. So we can make a few interesting observations here. At this high numerical aperture, confocal microscopy obtains optical sectioning, which originates from the curvature of the Ewald sphere. Furthermore, a point scanning OCT obtains a square root of 2 resolution enhancement over full field OCT, as we would expect for confocal gating. So now we discuss a limited depth of field or depth of focus in OCT. So as many practitioners of OCT know, OCT exhibits a limited depth of focus, which becomes more severe the higher the NA. But this seems contradictory because the concept of a transfer function or point transfer function assumes shift invariance. So if there's limited depth of focus, then that means there's a Z dependence. So about 15 years ago or so, some researchers invented interferometric synthetic aperture microscopy, or ISAM, which addresses this issue, which I will now discuss briefly. So the reason why we have limited depth of focus in OCT is that conventional OCT processing ignores this Ewald sphere curvature. And the penalty for this ignorance becomes more severe the higher the NA, because the curvature becomes more significant at higher NAs. So conventionally, this is what we assume, and we take 1D Fourier transforms along the wave number dimension, and then we get the limited depth of field. But if we resample this along the Ewald sphere, and then take a 3D Fourier transform, then we get the space invariant resolution. And this is the idea behind ISAM. This method does require, however, a phase stability during the full data acquisition, which means that the sample doesn't move significantly as you're, for example, sweeping the wavelength. So it turns out we can also explain Speckle using the bandpass nature of the transfer function of OCT without involving multiple scattering or strong assumptions about the sample. So in particular, if we start with the OCT image formation model, which is very much a coherent model, with and it's the convolution of a complex valued point spread function with a complex valued sample. And I'll note that this is not the same as convolving the intensities, which corresponds to an incoherent model. And the reason for this can be seen if we look at the functional form of this point spread function under the Gaussian assumptions that I made earlier. So we have so we're assuming the low NA limit, so they're separable into the axial and lateral components. And you'll note that both have this Gaussian shape, but that the axial component also has this complex modulation term that basically is the reason why this inequality doesn't hold. This equality doesn't hold. So let's start with a simple case of 2 closely spaced scatterers within your point spread function. So they have some axial spacing dz and some lateral spacing dx. And here I'm dropping the y dependence because I'm assuming the separability conditions, and therefore y behaves the same as x. So if we expand this responsive OCT, we get the incoherent terms that you would get as if you're using an incoherent imaging system. But you also get this coherent term, which has this modulation factor that only depends on the axial separation. And I argue that this is what contributes to the speckle that we see in OCT. And note that this coherent term goes to 0 when either the axial or lateral separation becomes larger than the resolution. So that means that these points are spaced further apart than the width of the PSF. And that's indicated by this factor here. And this makes sense because then they're no longer close to each other and they can no longer interfere with each other. So here's a comparison of the coherent versus incoherent model for two closely axially spaced reflectors, as denoted by these vertical bars. So the incoherent terms that you would get, you had an incoherent low-pass point spread function. And on the right, I'm plotting the same thing as on the left, but plotting the horizontal axis, the separation as the horizontal axis. So you note that as the reflectors get close together, you get this high frequency modulation for the coherent model, but you don't get that for the incoherent model. And in fact, it's even possible for two reflectors to be invisible under OCT, as indicated by the nulls in this plot. So one technique for reducing the speckle is incoherent angular compounding, which is also what we use in the OCRT for resolution enhancement, as we will see shortly. So if we just analyze that cosine factor that I mentioned earlier that's contributing to the speckle and examine the effects of angular compounding over a full 180 degrees or pi radians, then this evaluates to a Bessel function of the first kind. And here I plot a comparison between this Bessel function and the cosine. So the Bessel function will decay slowly, while cosine obviously goes on indefinitely. So this means that you only get that speckle modulation over the length scale of 1/k0, or the wavelength scale, rather than the width of the PSF in conventional OCT. So in this sense, angular diversity synthesizes incoherence over a larger domain. So if we add angular compounding to the previous comparison, we see that so here I plotted it in green. We see that this result is a lot more similar to the incoherent model. And again, I'm plotting out the video frames horizontally here. So there's still some modulation left at close separations, but by and large, it looks a lot more similar to the incoherent model. So this is how speckle reduction reduces, angular compounding reduces a speckle. So it might seem strange that I'm saying that two reflectors can give rise to speckle, but that's basically the base case. So we can go to a more general model of n reflectors inside your point spread function. So this right here, we have a superposition of n responses to n scatters. And this is basically a random phasor sum because this is a complex valued point spread function that I mentioned in that exponential factor a couple slides ago. So if you expand it, since this is a random phasor sum, in the limit of a large number of phasors, we get Gaussian statistics and therefore a fully developed speckle. So if we expand this expression, we get a similar result to before. So we have the incoherent terms, and then we have the coherent interference terms. And this is basically just the pairwise interference version of what we saw earlier. So we basically just simulated a general OCT intensity image. So it would be interesting to see the k-space coverage of this intensity OCT image that we have been analyzing. So we take the Fourier transform of this expression. So I of xz, and we get I twiddle of kx kz. So we get this very large expression that looks scary, but it's not as bad as it looks. So first thing I'll note is that it factors nicely into a product of what looks like a low-pass transfer function, which is the Fourier transform of the magnitude squared of the point spread of the coherent point spread function. And in the brackets on the right, we have basically the sample. So if you look at this sum, this is a superposition of complex exponentials, which if you take the Fourier transform of, you get a sum of delta functions corresponding to the reflector or scatter positions. And all of this is just interference that contributes to speckle that will get averaged away if you do angular compounding. So this will be, so in summary, if we assume that the sample is a sum of delta functions, then we can treat the transfer function governing intensity OCT as a low pass filter rather than a band pass. And this is important when we analyze OCRT, which is my next topic. So that's all I plan to discuss about this K-space theory. I had to cut out a lot. I just wanted to note that my paper discusses many other topics listed on this slide. And if you're interested, I'd be happy to chat offline or refer to you to the paper when it comes out, hopefully soon. Okay, so that concludes the first part of my talk. Hopefully it's clear now how we can obtain 3D information from wavevector diversity, which is equivalent to saying angular and wavelength diversity. So on this note, I will now talk about incorporating extreme angular diversity in OCT on top of its already high wavelength diversity. In particular, a technique that we've invented called optical coherence refraction tomography, or OCRT, and an extension, spectroscopic OCRT, or SOCRT. And I'll note that these two works have led to 2 peer-reviewed publications, one in Nature Photonics and one in Optics Letters. Okay, so the initial motivation of this work is that it's very often the case that the axial resolution of OCT is better than its lateral resolution. So in particular, in conventional OCT, while the axial resolution can be less than a micrometer if you have a large enough bandwidth, in practice, if you want to get millimeter scale imaging depths, you have to trade off your lateral resolution due to the trade-off between the lateral resolution and depth of focus. This typically means people accept a lateral resolution on the order of 10 microns or greater. So this is a well-known trade-off that's been a subject of intense investigation basically since the inception of OCT, and even before OCT. There have been many techniques that address this problem. For example, ISAM that I mentioned previously, which performs digital refocusing, and then there are a slew of beam shaping techniques that, for example, use Bessel beams, which theoretically have an infinite depth of focus, but with other trade-offs. So this list is not meant to be exhaustive, but rather illustrative, showing that these trade-offs are of significant interest in OCT. So here's where OCRT comes in, which is a technique that uses angular compounding to increase the lateral resolution of OCT to its axial limit, or otherwise remove any isotropy in the resolution. So as a consequence of the previous analysis, OCRT obtains Speckle reduction, and also because of the apparently low pass transfer function of intensity OCT images, we can now better appreciate how OCRT obtains resolution enhancement, which I'll explain shortly. And here, because we're combining the multi-angle images incoherently, it does not require phase stability. So to give a more concrete explanation, we start with this apparent low pass transfer function of intensity OCT images and the corresponding pointside function. So through incoherent multi-angle compounding, OCRT obtains a larger isotropic transfer function and therefore an isotropic pointside function given by the original axial resolution. So we get this sort of improvement over OCT. So everything is fine until we think back to our introductory optics courses where we learned about Snell's law. So basically, due to refractive index variation, which for biological tissue ranges from roughly, say, 1.3 to 1.5 or so, there will be significant ray bending, stretching, and compression. So for example, this is an OCT B scan or image of a circular tube, but due to refraction, the shape has become noticeably egg-like. So therefore, if we naively try to combine the multi-angle images, the B scans would not overlap and it would produce a large blurry mess. So thus we need to correct for these refractive distortions. And to do this, we use the ray equation as our forward model. which is basically the wave equation taken to the geometric optics limit. So the ray equation describes how a ray will propagate through some spatially inhomogeneous refractive index distribution given the boundary conditions, or in other words, the initial position and direction. So the ray equation will tell you how this trajectory will evolve in this through space. So these boundary conditions are determined in our case by the scanning geometry pictured here. So it depends on the lateral position as this beam is scanned laterally, and it also depends on the orientation of the sample, which we are rotating, at least in the first demonstration. And because there's no close solution to this, the ray equation in general, we have to use a numerical integration. So this all assumes that we know what the refractive index map is, which we don't in general, so we will need to optimize for it computationally. So I just described the image deformation model, which is the ray equation in this case. We now need some registration metric that specifies how well registered the B scans are. So we use an intensity-based, image intensity-based registration to guide the registration. So given these components, we optimize this registration metric with respect to the refractive index map until all the images are optimally registered to each other. And at this point, we know that the refractive index map is accurate. So at the end of the day, OCRT gives us two outputs. We get an isotropic and isotropic high resolution reconstruction here, and a co-registered refractive index of the sample. So here's an example of the progression of the optimization and registration over many iterations. If we start with a flat refractive index, we get a poorly registered image, but over time, they become more and more registered, and as the refractive index map becomes more and more accurate. So here's the initial setup we used to collect the multi-angle data. We inserted all the samples into glass capillary tubes with the help of a collaborator, Dr. Simone Deegan. The tubes were inserted or mounted onto a custom-built rotation stage, which was designed by Gar Waterman, a former lab member. And we use this setup to acquire OCT B scans or images every 6 degrees for a total of 60 orientations spaced across all 360 degrees. So at the end, we get this 3D data cube of XZ OCT B scans as a function of angle. And I'll note that for the 3D case, we would obtain 5D data cubes, basically 3D volumes as a function of 2 angles, which we will see shortly. So first, we did some quantitative validation experiments to show that OCRT can indeed obtain resolution enhancement and quantitative refractive index estimation. So to do this, we embedded a phantom sample consisting of sub-resolution polystyrene beads in 2% agarose, which has a similar refractive index to that of water. So this figure compares conventional OCT on the left and OCRT on the right. If we zoom into smaller regions, we can see the nice resolution enhancement, lateral resolution enhancement of OCRT over OCT, likewise in this panel. So to quantify this, we segmented out these beads and fit them to anisotropic Gaussians and plotted the full width half maxes. So we can see that for OCRT, the resolution is isotropic of around 2.5 microns, corresponding to the axial resolution of the B scan. And since the original lateral resolution of B scan was around 8 microns, we observed a greater than a 3-fold resolution enhancement. The refractive index maps also match the expected value. So for this agarose sample, we get a refractive index close to water. And we also repeated the same experiment in PDMS, and we get a refractive index map close to that of what we'd expect for a PDMS. So next, we imaged several mouse organs and applied OCRT. So this one is of a mouse femoral artery that has collapsed. So the upper left panel shows the average V scan with conventional OCT. And this panel shows the OCRT reconstruction. This panel shows the reconstructed refractive index map And this one shows the histological cross-validation. So on the top, we have a traditional H&E stain section, and on the bottom, we have the a trichrome stain, which brings out, I don't know if you can see it, but this wavy internal elastic lamina. So if we look at some zoom-ins at OCT and OCRT, we can see that OCRT has much better resolution and reduced speckle compared to OCT. And in particular, we can start to make out the wavy internal elastic lamina that you see here, which is consistent with the trichrome stain, this internal elastic lamina. However, in OCT, due to speckle and low resolution, we can't see this, can barely see this, if at all. We also imaged this. So this is a distended mouse bladder wall. So the slide is in the same format as the previous slide, and we can see a significant improvement of OCRT over OCT. In particular, we can see that the smooth muscle layers nicely in the OCRT reconstruction, but we can't really see it that well in the OCT, conventional OCT. And in the lamina propria, we see some finer structures, connective tissue structures that we don't see in the conventional OCT. We also imaged the human cornea. So from these zoom-ins, we can see that the enhanced resolution and speckle reduction in the stroma, where the lamina structure, so we can see that the lamina structures are much easier to make out and identify than in OCT. However, the primary point I'd like to bring up is the refractive index map, whose estimated values are around 1.38, 1.4 or so. And these are consistent with values obtained independently by other researchers. So we can conclude that OCRT can quantitatively obtain refractive index values. So next, I'll talk about an extension of this work to spectroscopic OCT, or SOCT. So in spectroscopic OCT, the anisotropy of the resolution is typically reversed, where the lateral resolution can be superior to the axial resolution. So this anisotropy in SOCT derives from the spectral axial resolution trade-off because of the time frequency analysis required in SOCT, such as a short-time Fourier transform, which forces an inverse relationship between the axial and spectral resolution of SOCT. So for example, you can use a relatively broad spectral window on the OCT interferogram, and then you would get decent axial resolution, but you would get poor spectral resolution because that's defined by the width of this window. So conversely, if you try to narrow down this window, you would get nice spectral resolution, but now you've traded off your axial resolution. So thus, when SOCT obtains good spectral resolution, which is the point of SOCT, the axial resolution must suffer and often becomes worse than the lateral resolution. So this means that, this presents an opportunity for OCRT to recover the axial resolution from the lateral resolution. And we call this technique spectroscopic OCRT, or SOCRT. So we demonstrated SOCRT on a mixture of 4 and 6 micron diameter beads embedded in 2% agarose. And we chose these beads because we know what the expected backscattered spectrum would be based on me theory. And it would also be difficult to differentiate them based on physical size alone because these are smaller than the system lateral resolution of around 8 microns. So if we do SOCT processing with enough spectral resolution, we get an axial resolution that's nearly 40 microns, which is 4 or 5 times worse than the lateral resolution of our OCT system of around 8 microns. So then we do SOCRT processing to recover the axial resolution. And we note that the spectral fidelity of the beads is not compromised. And they match very closely to each other, and they match very closely to the expected predictions by me theory. Okay, so for the last part of my talk, I will introduce a new class of methods we developed for multi-view imaging from a very wide angular range across 2 rotational axes without using a rotation stage or a glass tube that we had to use in this first demonstration. So I will then present how we use this new method for performing OCRT in 3D. So I'm actually presenting this work for the first time ever, which are the subjects of these two papers that I am currently working on. So the goal here is to expand the applicability of OCRT. So while we've obtained some impressive results in our previous setup, it has some severe limitations that limit its general applicability. So first of all, we had to cut up the samples and insert them into tubes, which obviously precludes any in vivo or imaging of live samples. This is further complicated by the fact that sample rotation is slow. And finally, because of the tube geometry, we can only rotate along one axis, which means we can only enhance one of the two lateral resolution, only one of the two lateral resolution dimensions. So we wanted to devise a method that addresses all of these concerns. And our solution was to use conic section mirrors, such as a parabolic mirror or an ellipsoidal mirror. So as we can see from this diagram, they can obtain extremely high angular coverages, theoretically even up to 4 pi steradians in the limit of an infinitely large parabolic mirror, for example. They're also well known to be perfect focusing elements. And I put this in quotes because I'll qualify this later. But nevertheless, parabolic mirrors are very rarely used for imaging and are typically more commonly used for efficient light collection or collimating divergent sources in the case of parabolic mirrors. So if they are perfect, why aren't people using them for imaging? And the answer is that they are only perfect on axis. However, if we want to form an image with many lateral pixels, then we need to tilt the incident beam in the case of a parabolic mirror. So that's what's going on in this animation. So as you tilt the beam, the focus shifts. And if we zoom in, we can clearly see, yeah, the focus is shifting, but the focus is only perfect when it's on axis, and then it rapidly deteriorates as you go more off axis. So this means that the field of view should be very limited. But how limited is this field of view, and what determines this field of view? So after a lot of math that I won't go through here, I derived these relatively simple equations that describe how the field of view varies as a function of a variety of system parameters, such as the wavelength, focal length, numerical aperture, and the radial entry position. And I verified these simulations in Zmax. And interestingly, these equations, I derived these for parabolic mirrors, but they interestingly also apply to ellipsoidal mirrors. So the most important takeaway from these equations is that the field of view is inversely related to the numerical aperture. So this means that we can reduce the numerical aperture to get larger fields of view. And this is exactly what we want to do in OCT anyway, because if you recall our discussion on the depth of focus versus lateral resolution trade-off, we want to use weakly focused beams. So this is actually perfect for OCRT. So I modified the OCT system so that the sample arm is now mounted on a 2D translation stage. And it's scanned across the aperture of a half parabolic mirror, and thus varying the angle of incidence. So although for this first demonstration, stage scanning is slow, we note that it is straightforward to achieve the same setup using a pair of anti-conjugate galvanometers or galvos. And so with this setup, you can rapidly acquire the OCRT data set. So this setup opens up the possibility of in vivo imaging of exposed surfaces such as cornea, skin, or endoscopically accessible mucosal tissues. So then we use this setup to capture 5D data sets of various samples for 3D OCRT. So in other words, we're capturing 3D volumes as a function of two angular dimensions, so X, Y, Z, KX, and KY. We acquired a total of 96 volumes, and we were able to achieve 1 millimeter squared fields of view over up to plus or minus 75 degrees, which you can appreciate if you look at, for example, this panel here, and maybe this panel here, which have nearly perpendicular views to this panel over here. And these are color coded based on the angle of incidence, by the way. that I've in this plot over here. So now we applied this 3D OCRT algorithm to a variety of samples, starting with fruit fly head. I would like to thank Dr. Ryan McNabb for generating these very nice 3D renderings. So on the left, we have the OCT volume, which has been averaged with the same number of frames as there are multi-angle volumes that went into this OCRT reconstruction. And because of the lateral resolution and speckle reduction by OCRT, we can better resolve the individual lenslets of this fly's compound eye, as well as some structures on top of the head. Furthermore, the antenna and the hairs are more finely resolved by OCRT than by OCT. So we also imaged zebrafish larva at 2 days post-fertilization, so it's 2 days old. And here I'm showing 2 fly-throughs of the 3D reconstruction. So on the left, we're flying through in Z, and I'm showing en face XY slices. And on the right, I'm showing, so we're flying through Y. And so this is basically an OCT-B scan. So let's start with the left comparison. We can see that OCRT has much better resolution and reduced speckle, which allows us to visualize this particular egg yolk structure in this developing zebrafish better than in OCT. And we can also see some structures in the brain or the head, as well as in the eye. Similarly, if we look at this comparison, we can see some structures that we don't see in conventional OCT. For example, let's see if I can. If I pause this video, we can even see the optic nerve and the retinal layers of the zebrafish's eye that we don't see at all in the OCT image. And if we compare this to histology, we see this corresponds very nicely. We have the optic nerve and then the layers of the retina, which in a zebrafish apparently, I guess there's no vitreous in a zebrafish eye. And we can even pause this video and you can actually see the optic nerve right here very nicely that you can't see in OCT. Yeah, so that was pretty cool. We also imaged the mouse esophagus. This is a cross-sectional view, so Z is a vertical dimension. So you'll note that we can see 2 different layers, these 2 outer muscle layers, because the fibers, as you're flying through, are moving in different directions that we don't really see in regular OCT. And we can also see the epithelium nicely in OCRT that we don't see in OCT. And this is all consistent with histological comparisons. So here we can see the 2 outer layers of the esophagus. In particular, it is well known that the esophagus has, the outer layer is longitudinal, so it goes along the esophageal tube. And then the internal layer is circumferential, so it wraps around the tube. And that's why we see these 2 different orientations going on here. And then we also see that the epithelial shape matches very nicely. So this is the same sample, but now I'm flying through normal to the muscle layers. And we can actually we can see when the layers switch, when the orientation of the fibers switches. Whereas for OCT, it all just looks, we don't see this transition very clearly at all. Finally, we imaged a mouse trachea. So here are the flying through Y and flying through Z on the right. And we can start to see these cartilage plates in the trachea that we don't see very clearly in the OCT, conventional OCT. So for example, if we pause this video around here, we can see, oops, pause it here. We can see this cartilage plate that matches closely with what we see here, as well as the epithelium on the bottom. And likewise here as well. we can see the cartilage plates and the epithelium. Yeah, so in summary, I've covered a unified case-based theory of OCT and new methods of incorporating extreme angular diversity in OCT. Yeah, so that concludes my talk. I'd like to acknowledge my committee members, who have been supportive during my graduate experience. I'd also like to thank members of all the people that I've worked with or offered guidance throughout the years, including people from the IZ lab, past and present, Warren lab, Farsi lab, and the Horsemeyer lab, and several other professors that have been extremely helpful. Yeah, so thank you all for listening to my talk, and I will take any questions you may have.
Please express yourself with icons or audible clapping, whatever you'd like. This is awesome. So there's a bunch of people here, including several special guests who this is the magic of Zoom. So several of our lab alumni, I see Chateau is actually on screen there. Frank's here. Someone named Francesco. I hope that's our Francesco. If not, I'm not sure who it is. Everybody, please feel free to just speak up with your questions, and let's go.
I guess it was a lot of information packed into 45 minutes or 50 minutes.
Oh, I was going to wait to get some other questions from students or co-workers, but might as well get something started. When you go back to the 3D OCRT image that you have of the fruit fly, the nice sepia-colored three-dimensional model, can you put that slide back up?
Oh, it is our Francesco. Hey, Francesco.
This one?
Yes, this one. Okay, so there is, it's obvious that there's most of the time there's spectacular improvements in the imaging, but you can certainly see in the OCT image something sitting at the left-hand side that's a branched dendritic structure, which doesn't show up in the OCRT. No, not that one, the one that's sitting farther up near the left eye. That right there.
This?
Hold on, right there.
Yeah.
I'll
Okay.
just pause it. Is this a good view?
So is that real? And if so, why doesn't it show up in the OCRT?
This part you're referring to, right?
Yes.
I think it is there. It's just that it just looks larger. It's supposed to be a fine structure. And because of the worse resolution of OCT, it just appears larger. I think it should be in the OCRT reconstruction. And if not, it might be because of the transparency. It might just be the way that it's being displayed. These antennae are most certainly, they are in the reconstruction. I think maybe we just need to work on this 3D rendering, make sure it's not hiding any features. So yeah, thanks for that feedback. That's good. Good to take.
Ryan might actually want to address that. We've discussed that several times, and Ryan did the rendering.
Yes, I need to go back and fix that. It's a mixture of, as Kevin said, transparency, and also the mask that was used to highlight all of that. It's all there, but I need to go. I haven't had time to go back and fix that.
Okay, that's
my
fine.
fault. I'll take responsibility for that.
Okay. And I did want to ask sort of a general question in the first part of your thesis, talking about the review article. It was extremely difficult to figure out from your presentation which aspects of that article are things that were known in the past and which ones are your own contribution. Can you just give me a quick summary of that?
Yeah, I think so. It was published as a review article, so most of it is not new. I think the insight is the insight that we brought in was tying it all together into one coherent, excuse the pun, one coherent framework.
Okay.
Although I will point out that I think that the speckle reduction analysis is new. As far as I can tell, I couldn't find any other treatments on that in the literature. I mean, one that was kind of similar, but not quite the same.
Okay.
And also the ISAM, I think ISAM was demonstrated for point scanning OCT and full field OCT, but interestingly not for line field OCT. So it seemed like a missed opportunity, but I think we were the first to do that as well.
Okay.
Do we have some other questions from the audience?
I have a question. So I think so. Can you you in your like in your last project you made a point about how the conic mirrors are really well suited for OCT, and I was wondering, hoping you could repeat that. I didn't get it the first time. And yeah, I was hoping you could just go over that again.
Yeah, so I think conic mirrors are ideally suited for OCRT because these relationships show that the field of view is inversely related to the numerical aperture. So if you want to get a large field of view, you have to decrease your numerical aperture, but OCT does this anyway to get long depths of focus. So it seems like it is ideally suited for OCRT in that regard.
Oh, I see. Okay.
So we got like millimeter fields of view with decent focusing, with lateral resolution of maybe 10 to 12 microns.
I see. And so when you started using these mirrors to like scan your sample at like many different angles, I'm assuming you're collecting like a lot of data, right? So yeah. Like what type of reconstruction times are you working with?
Yeah, so that's I didn't mention that. It takes maybe several hours, but maybe on the order of maybe 6 to 8 hours.
For one Drosophila head?
Yeah, for 1 millimeter by millimeter cube.
I see.
So we'll still need to work on that, but.
Okay. Yeah, cool, thanks.
Yeah.
Hi, Kevin, this is Ron. Very cool. So I don't know that much about OCT or OCRT, the optical kind of optics, but with the increased resolution in OCRT, I wonder the possible you can actually resolve single cells, individual cells, yeah, I think in the whole field of spatial omics, it's a very hot topic whether or not you can do spatial kind of 3D spatial single cell mapping.
Yeah, so I guess if you want cellular level detail, you'd want resolutions of like a micrometer or so. So our OCT system had an axial resolution of around 2.5 microns. So, but there have been OCT systems that sub-micrometer resolution. And if we had that system, then yeah, then we could probably do some start to resolve subcellular features. Yeah.
Mm-hmm. Well, is it possible to also integrate with like a fluorescence imaging, like a light sheet microscopy? They can look at a couple of fluorophores.
Yeah, maybe that's possible. And the contrast mechanisms are different, so that would be a good idea to sort of they would offer complementary contrast mechanisms. So yeah, we'd have to think about how to incorporate light sheet microscopy into this setup. And maybe it's possible.
It's cool.
By the way, this is Professor Fan. He was a professor I had at Yale, and yeah, I took a couple classes with him.
Yeah. Nice meeting you guys.
Wow, nice to meet you. Zoom defenses are amazing.
Am I going to be the last one in our?
I don't know. This is so great. We should probably keep doing it. Francesco, you have anything?
No, I think overall this is like very great. I saw a look at the paper from earlier. I think Frank showed me, and this is I think amazing work.
Thank you.
Yeah, I think there's a lot of promising applications here.
Oh, I don't have anything. I'm just glad I got a chance to work with Kevin in the past years.
Oh yeah,
Okay.
Frank was very helpful.
Seconding Frank, in this past few years that I had the opportunity of sitting in the meetings with Kevin, the feeling that I had, I guess for the first time I felt how the court musicians, when baby Mozart was playing, were feeling when they were seeing him growing and just doing this amazing job. So it's been just pleasure hanging out with Kevin.
Oh, thank you. You're too kind.
I will say, Kevin just did something which is one of the boldest things I've ever seen in a defense, which is he showed data that his advisors have not yet seen, which is an extremely risky thing to do. I think it worked out well because I finally believe the esophagus data, Kevin. But wow.
Was that the rotating muscle?
It's the esophagus tube.
Yeah, with the
cross muscle layers.
Yeah, that was fantastic.
Any other? Sorry to pick on those people in particular. Any other questions from the other attendees? Yeah, right here, Kevin. I finally believe that's a tube. The previous slide, and now I understand this one. Okay, well, thank you all so much for attending. This has really been special to see everybody. The committee members will stay on and we'll excuse the rest of the audience. So thank you very much for coming.
Thank you.
I would have one final question if it's allowed.
Oh, of course. Yes, please.
Oh, yes.
Please go ahead.
I saw the talk was recorded because I was a bit late. I'm in Germany. I was working late. Is it possible to see the talk?
Yeah, of course.
Somehow? Great.
Yeah, of
Then
course.
It'd be great if you could send me a link. Congratulations, by the way.
Oh, thanks, Lars.
Probably too early, huh? So I should.
Yeah, not congratulations yet. We got a lot of hard discussions to take place.
Okay, then I wait.
Okay, thanks everyone for joining.
Okay, we'll stop the recording now.